All right, thanks for watching this talk. I'm Amy ZimmerFaust, and I'll be discussing research that was conducted with the EPA in Newport, Oregon, looking for patterns and sources of nitrogen and fecal pollution in estuarine watersheds. Elevated nutrients and fecal pollution often co-occur, with problems from both of these stressors being widespread for a variety of watersheds. Elevated anthropogenic inputs of nitrogen can lead to eutrophication and low dissolved oxygen, with non-point nitrogen sources being a leading cause of water quality impairments in the US. Fecal pollution is also a common water quality impairment, causing costly closures of recreational and shellfish harvesting waters and public health risks to recreators. Sources of both nitrogen and fecal pollution can be difficult to manage due to the diversity of sources in watersheds with mixed land uses in particular. By understanding if and when sources co-occur, this can lead to bigger improvements and application of more strategic mitigation actions, which was the goal of these efforts. So I'll be talking today about a case study that was completed in the Tillamook Bay watershed, utilizing a multi-pronged approach to evaluating sources of fecal pollution and nitrogen in parallel and the potential co-occurrence of these sources. Tillamook Bay is a productive rural watershed on the Oregon coast that supports dairy, timber, and shellfish aquaculture. It also has a history of water quality impairments, including for temperature, dissolved oxygen, likely related to elevated nutrient levels, and fecal bacteria. Potential pollutant sources in the watershed include agriculture. There are extensive dairy operations in the lowlands. Tillamook cheese probably rings a bell. There's also wildlife and potential human sources, including both municipal and on-site wastewater treatment systems. This multitude of sources is not unlike what we see in other places, and it makes nutrient and fecal pollution management really challenging. So for this study to evaluate sources, samples were collected at 16 sites from the 5 major tributaries within the Tillamook watershed, approximately monthly over a 1-year period, and sites are shown with the black dots. So several methods were used in parallel to identify different sources of nitrogen and fecal pollution. Water samples were filtered and processed for microbial source tracking genetic markers using qPCR. Q, ruminant, dog, bird, and human fecal sources were all targeted using the markers shown on this slide. To tease apart sources of nitrogen, we did a few things. From the same sample, we took the filtrate and measured nitrate stable isotopes and nutrients. Nitrate stable isotopes are really useful for teasing apart sources of nitrate, and enrichment of the isotope is associated with anthropogenic sourcing, including inputs from cows or human. So the idea was that by pairing the different source tracking methods, we would be able to tease apart some of the more complicated trends in nitrate and pollution sources, like inputs from cows versus humans. So we made all of these measurements on the water samples, and then in parallel, we used the Sparrow watershed modeling tool to further characterize our sites. The Sparrow model predicts the total nitrogen load at each site and attributes it to different sources, with the predicted contributions based on land use, monitoring data, and watershed characteristics that may impact pollutant fate and transport. So to recap, we used nitrate and fecal pollution source tracking tools in parallel, and then we further characterized our sites by likely nitrogen sources with the Sparrow watershed modeling tool. So I'll start here by briefly going through what just the MST marker results tell us. So here we are looking at the watershed sources of fecal pollution. I'm just going to highlight results from a few of the markers measured. Here on the x-axis, sites are shown moving along a downstream gradient, and each tributary is shown with a different color. So first, we'll just look at the ruminant marker on the left to start. One thing to note is that the ruminant marker was highly correlated with both cow markers. So we'll just be talking about the ruminant marker, but it's reflective of those cow marker trends too. So we see that the ruminant marker is frequently detected, and that concentrations increase moving downstream as we move from more forested to more agricultural land uses. So the takeaway is that cow and ruminant markers were detected in high regularity in all 5 of the rivers. In contrast, we just detected the human marker at high concentrations in one of the tributaries, and we didn't see trends moving downstream. We saw instead that the elevated levels were linked to a specific area, which was adjacent to a wastewater treatment plant. So we also looked for relationships between the markers and E. coli. Here are just the wet season trends, but we saw similar things in the dry season. And we detected significant relationships between ruminant and cow markers and E. coli levels. So we have strong evidence from the MST results that cows are a primary source of fecal pollution at most sites within this watershed. So next we looked at sources of nitrogen to the system. First, we looked at trends in the nitrate stable isotopes, again along the same downstream gradient, which is on the x-axis. And here again, the different tributaries are different colors. What we saw is that as sites move downstream from forested to ag lands, we see enrichment of the nitrate stable isotope signature. The increases we see here reflect non-natural sourcing of nitrate moving downstream, but from these results, we can't differentiate between human versus cow waste as the likely source. So next, we'll just look at one of the rivers, the Trask, which was the blue line on the left, on one day. But these trends are reflective of what we saw consistently over the course of the study. Here we overlaid cow marker results with the nitrate stable isotope results. We see really similar increases moving downstream. So looking at these results together, this starts to be suggestive of cows as a primary source of nitrate and a likely reason for the enrichment in the nitrate stable isotopic signature. So the second way we thought about nitrogen sources was with the Sparrow watershed modeling tool. For this piece, we were interested in how well the Sparrow model site characterizations matched with what we saw with both the nitrate stable isotopes and the MST markers. The Sparrow model results will give us a percent of the total nitrogen load attributed to different sources at the different sites. So here on the x-axis, I'm just showing the predicted percent total nitrogen estimated from manure and fertilizer for each site. So this is the predicted ag-related nitrogen inputs from the Sparrow model. On the left, we see the strong relationship with the nitrate stable isotope signature in green, and on the right, we see a strong relationship with the ruminant marker as well in blue. So even though the Sparrow model is predicting sources of nitrogen, in this system, it was still a good predictor of both enrichment of the nitrate stable isotopic signature and what we saw with the MST markers. So finally, we can put all of the methods together. Here on the x-axis, this is from the Sparrow watershed model. Again, this is the percent total nitrogen estimated from manure and fertilizer. On the y-axis, we have those nitrate stable isotope results. Then the color in the sides of the dots, this is showing the MST human and ruminant marker results. What we see is that with increasing ruminant marker levels, so as we move to the bluish colors, you see increasing nitrate stable isotope signature, indicating anthropogenic sourcing of nitrate, and increased ag nitrogen inputs estimated from the Sparrow model results as well. So there are these two outliers that veer off the line here, where we do have higher nitrate isotopic signature than expected from just the predicted ag inputs. If you remember, the size of the circle indicates the human marker results. So these are the two sites where we frequently detected human marker at elevated levels, and these sites are adjacent to that wastewater treatment plant. So taken together, we can conclude that at the majority of the sites, all the sites along this line, ag inputs are the biggest contributor to both sources of nitrate and fecal pollution. But we were also able to identify sites where a specific point source is also impacting water quality. So for lessons learned, this combined approach that we applied, and by looking at multiple co-pollutants together, this really allowed us to tease apart these more complicated source patterns. The Sparrow watershed modeling tool also did a good job at characterizing what sources are problematic at specific sites. Now, even though this is a model designed for nitrogen, rather nutrients, it was still a good predictor for trends in the MST marker results as well in this watershed. So this study really suggests that nitrogen and fecal pollution are entering the Tillamook Bay as co-pollutants. The next question will be focused on how they are entering so that mitigation efforts can be further targeted. So with that, I'd like to thank all the folks and groups that contributed to these efforts. This was really fun and very collaborative project. So thank you.